Hello, everyone. So first off, the eternal question, why long reads? My lab originally got into long reads because we are a splicing lab, and long read sequencing allow us to sequence the entire transcript in a single read and provide the full transcriptional context of alternative splicing instead of understanding it a single splice junction at a time. And this allows us to do much more functional prediction and such things. However, we've now kind of realized the potential of this technology to understand all of these different types of transcriptomic alterations that can lead to cancer. And we can assay them all with the sequencing method. And so first I want to talk about detecting alternative splicing and isoforms and expression. And for this, we developed a tool called FLAIR that takes your aligned reads and identifies a high-confidence set of transcripts. And this can be really useful in different types of cancers. For instance, here's a study we did with human bronchial epithelial cells after a mutation in the highly mutated splicing factor U2AF1. And here we see that this gene, UPP1, has many different isoforms, but we're focusing on 5 of them here. In the wild type, we see a pretty even distribution of these 5 isoforms. But in the mutant, we see that these 2 green isoforms have much higher usage. And this is a result that is really well recapitulated in the RT-PCR. And this is really exciting because these isoforms are actually more productive. They make more protein. And that's a very interesting functional result for this gene. However, we've now updated FLAIR to incorporate more of this variation that you can see in the transcriptome, such as allele-specific expression and RNA editing. So FLAIR now integrates variant calls with the high-confidence transcripts to get haplotype-specific transcript reference. And what we can do with this is really exciting. So here is the mouse gene, MCM5, and we can see that it has 2 dominant isoforms, the blue one and the green one. And we can see that the blue one is from one parent and the green is from another. And these 2 dominant isoforms are differently spliced. So we're seeing differential transcript expression from the different haplotypes. And other work that we've done has shown that this can be relevant in cancer genes. However, there's other things that can really only be detected with long reads, such as RNA editing. So the ADAR enzyme is endogenous to our cells and changes adenosines to inosines, which can lead to splicing alteration, codon changes, and changes to the RNA structure. However, this is really difficult to study because these can be clustered changes. So multiple A's in a row can be changed to I's. And this is really hard to detect with short read sequencing. So here we can see that with Illumina, very few, only 1 in this sequence of A to I changes are detected. However, with Nanopore, we can detect way more. And we can show that with ADAR knockdown, we remove those changes, so this is not just an artifact of our process. And so moving on to another really exciting part of the cancer transcriptome, and that's gene fusion isoforms. So gene fusions are a really well-known oncogenic mutation that happens when there are double-stranded breaks in the DNA, and then DNA from different loci come together, and multiple genes are expressed under the same promoter. And so we can imagine that you could have a known cancer-driving fusion that is targeted by an existing therapy, but then it can be alternatively spliced. And if we don't understand this alternative splicing and take it into account when developing therapies, you could actually have isoforms that are resistant. And so I developed a tool called FLAIR-fusion, which identifies the gene fusions by identifying chimeric reads, and then after independently identifying the isoforms from each locus, combines them to understand the full gene fusion isoform. And so what I've shown is that the current best short read tools for gene fusion identification have very little overlap. Here we see that they only agree on 3 fusions, while long read tools for fusion identification agree on a much larger set of fusions, showing that they're more sensitive and consistent in fusion detection. Oop. And then what FLAIR-fusion does that's unique from all of this is the isoform identification. And so here we see two genes, PIWIL4 and GUCY1A2. And these two genes originally face in opposite directions on chromosome 11, and in this fusion, they're smushed together. And so we see that 13% of the locus is made up of the second isoform, which misses the, this exon here. And this second isoform is actually made up of a completely novel combination of junctions. So it's not seen in the reference. And this is a fusion that is seen in lung cancer. So we're continuing to look into this result. And then taking all of these tools that we've developed for understanding the cancer transcriptome, my most recent project is beginning to understand it in a single cell-based way. So for most short-read single-cell methods, they actually only sequence a very small portion of the end of the gene. So all you can understand is gene expression. With long reads, we can understand much more, including the fusions and the indels and the splicing. And so with that, we can begin to understand more about what drivers are emerging and what's changing on a single cell level. So to kind of expand on this diagram, we can see this is a model of a cancer. So you start with a normal cell that begins to accumulate mutations until you have a heterogeneous population. And then after a treatment, you end up enriching for specific mutations. And so our model in this experiment was using a PC9 cell line that is driven by an EGFR mutation, and then treating it with a tyrosine kinase inhibitor, which inhibits the EGFR mutation, forcing novel drivers to emerge. And so what we get is this UMAP of the single cell sequencing, where what we can see is that there's two novel clusters emerging in the treated cells in the upper right here. And so what we did is a global analysis of this transcriptome in a pseudo-bulk manner. So we looked for fusions, splicing changes, indels, everything. And we were able to recapitulate known mutations in this sample, but we were also able to find that our biggest hit is a known cancer driver, KRAS. And so we can see a few different exciting things in this data. So there is a increase in KRAS in this cluster and a decrease in this other novel cluster next to it. So there appears to be two different mechanisms here, as well as a increase in expression of the KRAS4A isoform globally across the treated samples. And so this is really exciting as well, because the KRAS4A isoform differs by the inclusion of this extra exon in the membrane targeting domain, and has previously been shown to be increased in tumor samples as opposed to KRAS4B, which is the dominant isoform and is unchanged in tumor samples. So we're continuing to follow up on this result and others in the single cell data, but I wanted to share this exciting result with you all. So thank you very much. I would like to thank my lab, particularly Angela Brooks, Cameron Soulette, and Allison all worked on this, some of these projects. And the Alice Berger lab did a lot of the single cell work with us. And they're amazing. They're at Fred Hutch. So yeah, thank you all for coming.